to another episode of Focus. Alright, there's a lot going on in the world as usual. I mean, Donald Trump has become president, Elon Musk has become the official minister or advisor of efficiency, whatever the title be. Doge sounds like his coin, but conflicts and power changing hands in Syria and Myanmar. And did you also see what Elon Musk or SpaceX managed to carry out? They caught a rocket. They caught a rocket. Come on. This place is amazing. This planet, fantastic. But you know what? We're here for something else entirely. As always, I'm always reading and going through, and I come across some absolutely amazing and fantastic stories. This one, I think, might be a follow-on or a play-on to a couple other stories I've covered, but it's an amazing breakthrough, nonetheless, in, say, medical technology, but that could have innovations or far-reaching ramifications in the wider industry or the world. Let's see. Stick around. Once again, welcome back to another episode of Focus, and thanks for joining. Scientists create an extraordinary mouse using genes older than the animal itself. Apologies. New research unveils the evolutionary origins of stem cells through a groundbreaking experiment that successfully created a mouse using ancient genetic tools. Published in Nature Communications, an international team of researchers has achieved a groundbreaking milestone: the creation of mouse stem cells capable of generating a fully developed mouse. This was accomplished using genetic tools derived from a unicellular organism with which we share a common ancestor, predating animals. This breakthrough not only reshapes our understanding of the genetic origins of stem cells, but also offers a new perspective on the evolutionary connections between animals and their ancient single-celled relatives. In an experiment that sounds like science fiction, Dr. Alex D. Mendoza of Queen Mary University of London collaborated with researchers from the University of Hong Kong to use a gene found in choanoflagellates, a single-celled organism related to animals, to create stem cells which they then used to give rise to a living, breathing mouse. Choanoflagellates are the closest living relatives of animals, and their genomes contained versions of the genes SOX or SOX and the POU, known for driving pluripotency, the cellular potential to develop into any cell type within mammalian stem cells. This unexpected discovery challenges a long-standing belief that these genes evolved exclusively within animals. By successfully creating a mouse using molecular tools derived from our single-celled relatives, we're witnessing an extraordinary continuity of function across nearly a billion years of evolution, said Dr. D. Mendoza. The study implies that key genes involved in stem cell formation might have originated far earlier than the stem cells themselves, perhaps helping pave the way for the multicellular life we see today, from single-celled relatives to multicellular applications. The 2012 Nobel Prize to Shinya Yamanaka demonstrated that it is possible to obtain stem cells from differentiated cells just by expressing 4 factors, including a SOX or SOX2 and a POU or OCT4 gene. In this new research, through a set of experiments conducted in collaboration with Dr. Ralph Jonch's lab in the University of Hong Kong, Center for Translational Stem Cell Biology, the team introduced choanoflagellate SOX genes into mouse cells, replacing the native SOX or SOX2 gene, achieving reprogramming towards the pluripotent stem cell state. To validate the efficacy of these reprogrammed cells, they were injected into a developing mouse embryo. The resulting chimeric mouse displayed physical traits from both the donor embryo and the lab-induced stem cells, such as black fur patches and dark eyes, confirming that these ancient genes played a crucial role in making stem cells compatible with the animal's development. Evolutionary roots of stem cell machinery. The study traces how early versions of the SOX and the POU proteins, which bind the DNA and regulate other genes, were used by our unicellular ancestors for functions that would later become integral to stem cell formation and animal development. Choanoflagellates don't have stem cells. They're single-celled organisms, but they have these genes likely to control basic cellular processes that multicellular animals probably later repurposed for building complex bodies, explained Dr. D. Mendoza. This novel insight emphasizes the evolutionary versatility of genetic tools and offers a glimpse into how early life forms might have harnessed similar mechanisms to drive cellular specialization. Long before true multicellular organisms came into being and into the importance of recycling in evolution, this discovery has implications beyond evolutionary biology, potentially informing new advances in regenerative medicine. By deepening our understanding of how stem cell machinery evolved, scientists may identify new ways to optimize stem cell therapies and improve cell reprogramming techniques for treating diseases or repairing damaged tissue. Studying the ancient roots of these genetic tools lets us innovate with a clearer view of how pluripotency mechanisms can be tweaked or optimized. Dr. Jauch or Jonch, apologies if I'm butchering that name, said, noting that advancements could arise from experimenting with synthetic versions of these genes that might perform even better than native animal genes in certain contexts. And like I always say, Earth is amazing. I mean, we would never know about any of this if not for the amazing people that inhabit it. Wouldn't you say? As always, as I like to say, the Earth is an amazing place. Not just the people, not just the places, but what we have done with it, what we can do with it, and what we have yet still to do with it. Thanks for joining for another episode of Focus. If you've got the time, stick around for the thoughts and facts section. But as always, have a great day, take care, and stay safe. But as always, if you're still around, thanks for sticking in and staying tuned for the thoughts and facts section. As always, you know how it goes. I give you a few facts and I lay out a few of my thoughts. In this case, I'm gonna lay out the facts for you and I'm gonna leave a trail. You might just get there before I do. So, let's get started. Choanoflagellates. Choanoflagellates are a group of free-living unicellular and colonial flagellate eukaryotes, considered to be the closest living relatives of animals. What is the function of choanoflagellates? By grazing on bacteria and detritus, choanoflagellates in turn feed their larger predators and return dissolved organic material to the water column. Both in number and feeding efficiency, choanoflagellates form a critical link in the microbial food chain that supports the global economy. ecology. Apologies. Funny guy. But the choanoflagellates are a group of, and I said this, having a funnel-shaped collar of interconnected microvilli or microvilli at the base of a flagellum. Choanoflagellates are capable of both asexual and sexual reproduction. They have a distinctive cell morphology characterized by an ovoid or spherical body that's about 3 to 10 in diameter, with a single apical flagellum surrounded by a collar of 30 to 40 microvilli. Movement of the flagellum creates water currents that can propel free-swimming choanoflagellates through the water column and trap bacteria and detritus against the collar of the microvilli or microvilli. Where these foodstuffs are engulfed. This feeding provides a critical link within the global carbon cycle, linking trophic levels. In addition to their critical ecological roles, choanoflagellates are of particular interest to evolutionary biologists studying the origins of multicellularity in animals. As the closest living relatives of animals, choanoflagellates serve as a useful model for reconstructions of the last unicellular ancestors of animals. According to a 2021 Crown Group Craspides and perhaps Crown Group choanoflagellates, if an Cothecidia arose with Craspedia, appeared 422.78 million years ago, although a previous study from 2017 recovered a divergence of the Crown Group choanoflagellates at 786.62 million years ago. And yes, I know I go on and on a little bit, but here's a little fun fact. Why are choanoflagellates important to our planet's health? Small as they are, choanoflagellates have a huge impact on the food chain, aiding to the flow of carbon from the primary producers to higher trophic levels, including ourselves. They comprise 5 to 40% of the heterotrophic nanoflagellates, and they filter between 10% and 25% of the coastal seawater or the coastal surface water each day. By grazing on bacteria and detritus, choanoflagellates in turn feed their larger predators and return dissolved organic material to the water column. Both in number and feeding efficiency, you get the picture. But as we go on, shall I go stem cells or pluripotency? Let's go stem cells, just as a refresher. Stem cells are the cells that can develop into many different types of cells in the body and are considered the body's building blocks. Stem cells are cells with the potential to develop into many different types of cells. They serve as a repair system for the body, and there are two main types of stem cells: embryonic stem cells and adult stem cells. How do you get stem cells? The most common way to harvest, or before this new study, the most common way to harvest stem cells involved temporarily removing blood from the body, separating out the stem cells, and then returning the blood to the body. To boost the number of stem cells in the blood, medicine that stimulates their production would be given for about 4 days prior to the actual procedure. So, tracking back, pluripotency. Pluripotency is the ability of a cell or substance to have multiple potentials or to differentiate into different cell types. These cells have the potential to differentiate into any cell or tissue in the body, including the three body layers: ectoderm, endoderm, and the mesoderm. The germ cells, embryonic embryonic stem cells, and induced pluripotent stem cells, or the iPS cells, are examples of pluripotent stem cells. And the POU genes. The POU genes represent a larger class of DNA binding transcription factors known for their roles in the cell type specification and the development regulation. Ryan and Rosefield 1997 and Phillips and Louisi 2000 publication. The POU homologue, or the OCT4, has been extensively studied and is the most critical of the 4 Yamanaka factors used to apologies, need a coffee break. Alright, that's better. Where were we? Right where the relevance was rearing its ugly head. Most critical of the 4 Yamanaka factors used to induce pluripotent stem cells in mammals, which had to be mentioned. The SOX and the POU are families of transcription factors that are crucial for regulating gene expression programs that determine cellular identities. They are involved in the development of the nervous system and are associated with stemness. SOX and the POU transcription factors are associated with stemness and were believed to be animal innovations, reported absent in their unicellular relatives, which was reported. Here they describe the unicellular SOX and POU factors, choanoflagellates, and the phyllostirine SOX proteins have DNA binding specificity similar to mammalian SOX2 genes, or the SOX2 genes. Now, I had to mention all of this. I'm missing one more. Yeah, one more. Because if you're like me and you know a little bit, or anyway, just to help you differentiate between the two, between the metazoa and the protozoa. Protozoans are simple unicellular organisms which are considered to be primitive types of animals, while metazoans, ourselves, are multicellular and are divided into, invertebrates and vertebrates. These multicellular organisms have cells that are specialized into tissues and organs, enabling them to perform specific functions. The difference between prozoa and metazoa. Sponges or porphyria, because of certain unique features of their organization, are usually regarded as a separate side branch from the metazoa. The parazoa, they have neither the true tissue nor organ characteristics of the metazoans, and their cells retain a considerable degree of independence. Now, I know I've bored you with a lot of the facts here, so before I even take you to my thoughts, I've just got one more thing that needs to be mentioned. What I have is, somewhere if I can find it, an excerpt or something I've highlighted from the actual study that was published. If you can just bear with me for a moment. I want to just give you a bit of the discussion, which I have lost completely, and the method they used to actually carry out this experiment. So, in the meantime, why don't you just enjoy the music? But as we proceed, here's just a short excerpt or highlight, as I said, from the discussion from the actual paper that was published regarding this experiment. In this study, we show that the SOX and the POU, two of the most important TF families associated with mammalian pluripotency, emerged before the origin of animals. activating stem cells in their multicellular context. SOX and the POU factors are therefore older than previously thought. The somatic cell reprogramming of mouse fibroblasts into the iPSC requires SOX proteins to bind and open-closed chromatin. The molecular capacity to bind and deform and open up closed chromatin as a pioneer factors and might have primed the SOX proteins for being deployed as regulators of diverse stem cells across animals. Now, you know what? I know I've been very technical and I've given you a lot of the science, so let me just break a little bit of this down for you. Basically, what they have discovered is this unicellular organism, protozoa, is basically like the on and off switch or the trigger that we can use to create or accurately drive what direction we would like stem cells to go into. Further, moving on that, in case you didn't know, right now the reality is that we can use stem cells to regrow certain things, body parts, not entirely, but I'm getting there, bear with me. Anyway, the molecular capacity to bind, deform, and open up closed chromatin as pioneer factors might have primed SOX proteins for being developed as regulators of diverse stem cells across animals. It will be of interest to test with direct data from early branching animals' lineages whether this pioneering activity is widespread across extant lineages. Our data shows that many of the biological or biomimical features of the mammalian SOX-B factors can be found in unicellular holozones and the reconstructed sequences predating the last common ancestors of animals. We propose that these features may have facilitated SOX co-option into gene regulatory networks of stem cells in unicellular holozones. A regulatory role of SOX factors in driving cell fate conversions might be less critical, which could explain the pattern of rampant loss of bona fide SOX across the surveyed taxa. Yet with the appearance of animals, SOX factors are retained across all extant lineages, indicating functional essentiality. Since many unicellular holozone species surveyed as transcriptomes or draft genomes, higher quality genomes from unicellular holozones might reveal SOX or POU sequences in more species. More broadly, our study shows that no single extant species can be used as the only representative of the outgroup of animals. That has wider implications. I'll get there. Frequent gene loss and millions of years of independent adaptation resulted in a mosaic of ancestral and derived traits in extant unicellular holozones, beautifully exemplified by the patchy distribution of the critical TFs. Even so, the remarkable conservation of function on the striking capacity of a unicellular SOX to direct the iPSC induction in mammals shows how the complexity of multicellular gene regulation has deep roots in our unicellular ancestry. The SOX family underwent several rounds of duplications in animals. Some newly emerging families might have lost some of their ancestral characteristics, exemplified by the appearance of pluripotency, incompetent ancestral sequences of the SOX, C, E, and F classes. We propose that the induction of the E57 was a critical step for the gene family expansion of animal SOX, as it allowed the SOX, C, E, and F members to diversify their partners and DNA targets. which basically means it allowed them the capability to actually become all of these other cells, i.e. our organs, our eyes, our nose, whatever you want to say. But allowing the partners and DNA targets, allowing non-overlapping regulatory networks. SOX factors retain K57 across animals, which we speculate might be critical for SOX-B to function in stem cells in cnidarians, sponges, cynophores, centophores, and placozoans. And I have no idea what that one is. Bilaterians, including planarian neoblasts. And that's as much of the discussion that's relevant. Now I'm just going to cover as fast as I can the method they actually use to do this. So just, to give anyone peace of mind, the ethical approvals. The animal study protocols were approved by and conducted in compliance with the Committee on the Use of Live Animals in Teaching and Research, or the CULTA reference number 22 to 276 at the University of Hong Kong, and the Animals Control of Experiments, Ordinance of Hong Kong, and Alright. The generation of a chimeric mouse from choanoflagellates, the SOX iPSCs. Now, I'm going to read out a couple of numbers and things. This is only to give you, if anything, if you take the time to actually go and read this paper, it'll give you some direction towards the diagrams. So, just so you know, I haven't lost it quite yet. The generation of a chimeric mouse. The OG2 MEF cells were derived from day 13.5 embryos of an OG2 mice or a mouse in Jackson Laboratory number 0044654. Following the preparation protocol described previously in reference 53, colonial iPSCs derived from the OG2 MEF derived from the CM7 These are just the different mouses or mices. Basically, there's a white one and there's a black one. So we go. The CN7BL black. J6 strain with the black coat color reprogrammed by full-length Sahel SOX I were cultured in a 2I LIF medium on a gelatin-coated plate for 7 to 10 days before they harvested and injected with a medium change every other day. The CD1 strain, or the white color coat from the female mice, were euthanized for isolating the moriola or the blastocytes using a 27G blunt end needle. These blastocytes were then collected and incubated in a K-SOM medium. I know I'm sounding crazy, but I'll get there. And until the injection of the iPSCs was performed on the day of the injection, 1 million cells were prepared to be selected for the microinjection into the blastocytes. The injection process took place on a 6 centimeter plate using a laser objective. A slit was created on the zona pellucida following with 8 to 10 selected iPSCs, which is basically each one of the genes, were transferred into each blastocyte using a microneedle. Following the microinjection procedure, approximately 25 blastocytes containing the integrated colonial iPSCs were implanted into the pseudo-pregnant CD1 mouse. They would be nurtured and allowed to develop in a conducive environment. This fostering and breeding stage is critical for the establishment of a chimeric mice derived from the colonial iPSCs, ensuring that the generation of genetically homogeneous and robust model organisms for further experiment investigations. Now, I don't know what to say. I know it sounded like a lot of science babble and science talk, but I can tell you this to those who don't know. To any scientists, I just literally told them how they could do this themselves. And it's amazing, right? So, just as we go, what this all is, what does it mean? Because I know I was sounding very, I don't know, far gone maybe. The reality of this technology, of this, how can you call it, new development, means it could actually change the game on what we know as not just stem cell development, but gene therapy. It can go a long way in regenerative medical or medicines, as they said, because what this has now enabled us to do, or given scientists or even doctors the capacity to do. Alright, let's rewind for a second before I get there. Remember way back when I did an episode on a company called Organox? Remember them? Yeah. They developed a fluid using a machine, but the fluid itself was the more, how can I say it, eye catcher. This fluid was capable of more or less reviving dead tissue cells, dead blood cells. This short study or short test that they did was actually capable of restarting or reviving a pig's heart and kidneys and liver, and it revived these organs with at least 40% or 50% efficiency. Now let's fast forward. We have got this new development which changes the, how you call it, what we now know as the use of genetic tools. I'm sure you've heard of what did they call it, CRISPR. But anyway, before we get there, you know I love innovations, integrations. I mean, that's where the world is heading at. So let's take a step back and let's look at this. The Organox technology, let's say, put together with this new enlightenment means that because I took a stab at what their mystery concoction of chemicals could be, and like I said, it had to involve some form of stem cell treatment or stem cell technology in there. So let's talk about this. What we have now with this study is the just give me a second. Okay, alright. I really hope none of that was picked up. And a lot of time just elapsed there. Apologies. But jumping back in once again. 2 technologies, innovation, creation. What can we do with it? The long and short, since I've already taken up way too much of your time and literally just given air. And most likely no one would have carried on listening if you see they heard all of that nothingness. Either way, let's get back into it. What we have here is the potential to create, once again, a formula if you want to use CRISPR, or a serum if you want to use OrganOx's method that can either create, repair, or regrow damaged tissue. Now, I know when I say it, it sounds crazy. And even the scientists themselves, when they said they carried out this experiment, it sounded like it was something out of sci-fi. But this isn't sci-fi. This is our reality right now. With the technology available in our hands on this planet. I mean, it is really, really amazing. Maybe only to me. It's just my opinion. But since I have elapsed and maybe digressed a few times, I'll leave you on this note. I know this much. There's another company, actually a few out there, that might be really interested in this new study. Have you heard of Colossus? No? They're the company that wants to bring back the woolly mammoth. I think they had a promotional video where they created either a mammoth steak or a saber-tooth steak. Heard about it? If you haven't, you should really check it out. By the way, did you know even Paris Hilton is actually one of the backers? And they're doing another round of funding? I mean, it's real. They want to bring back the woolly mammoth, the Tasmanian tiger, the dodo bird. Hey, Earth is amazing. Not just the people, not just the places, but what we can do with it, what we have done with it, and what we have yet still to do with it. This technology can literally change the world. The potentials are endless. But as always, as I like to say, our history is pretty vast and we're making it pretty fast. So, in the grand scale of it all, what was 41 minutes of your time? I appreciate every second that you had to spare, and thanks for tuning in once again to another episode of Focus. And as I always say, Carpe Diem. Let no one destroy your zen. Let no one destroy your peace. Take care.